Hello, I'm Jacob from the Cedar Oaks Public Library, and you're watching Adventures in Science, where I will attempt and explain various scientific experiments with varying degrees of success. Because if there's one thing you need to know about science, is that sometimes it's more art than science. So today, we are going to attempt to create color-changing flowers. Not these flowers specifically, I just thought that in case you hadn't seen a flower in a while, I'd remind you. Anyway, before we get to the color-changing part, we are going to need to get into a bit about how plants work. While animals have veins and use blood to transport nutrients, plants contain special vascular tissues known as xylem and phloem. Xylem is a firmer, dead tissue that transports water and minerals up, and only up, from the roots to the leaves. Phloem is a softer, living tissue that transports sugar molecules, amino acids, and even hormones both up and down through a mix of water and minerals called sap. Now, why did I tell you all that? Well, plants absorb nutrients and water through their roots, but the process where plants create their fuel, aka photosynthesis, actually occurs in the leaves. Naturally, that means that every plant needs to have a way to force their food from the ground up so that it can be digested and metabolized. Based on our understanding of xylem and phloem, we know water and nutrients can go up, but there's a tiny force called gravity fighting against it. So how exactly does a plant overcome gravity? Well, through a couple different ways actually, the culmination of which is called capillary action. The first step in capillary action is known as the force of adhesion, which in this case is the attraction between the water molecules and the plant fibers. The second is the force of cohesion, which is the attraction of one water molecule to basically every other water molecule ever. However, there is another process that helps plants move water through the xylem called transpiration. When water evaporates from the leaves, buds, and petals of a plant, more water is drawn upward to fill the empty space. Kind of like what happens when you suck water through a straw, only drastically weaker, and with a much smaller straw. In fact, it is estimated that 95% of water absorbed by a plant is lost to transpiration. Hypothetically, we should be able to see how these processes work using flowers, water, and food coloring, which should also allow us to create our color-changing flowers. In order to make this a little bit more of an experiment, we're going to test whether there is a difference in how different colors are absorbed in the plant, and also whether any sort of capillary action / transpiration is possible when the stem is divided in two and placed into two differently colored solutions. And if so, do different dyes in the water interact at all as they travel up the flower? We are going to start by setting up the whole stemmed flower portion of our experiment. First, we're going to pour about 3/4 of a cup of water into all of our glasses before adding a copious amount of different food coloring into each one. For these, we're going to test the colors red, blue, green, and finally, purple. Next, we are going to add our flowers to our variously colored solutions. You'll notice that I doubled up on the cups I was using. This is because they felt extraordinarily light, and the last thing I wanted is for them to fall over and dye my work table a kaleidoscope of different colors. Unless... No, never mind. Let's move on. Now it's time to move on to the split stem portion of the experiment. After carefully splitting 3 of the flower's stems off camera, we are going to add water to 3 pairs of glasses. The first pair we will dye red and blue. The second, yellow and red. And the final one, blue and yellow. These color pairings will allow us to easily see that if transpiration or capillary action is happening, whether or not the water from each cup is spreading to the other side of the flower. With all our flowers placed into their designated cups, we are going to come back and check on them at 4 predetermined points: 1 hour, 4 hours, 8 hours, and finally, 24 hours. After 1 hour, the whole stem plants in the blue and green solutions have already started to change color, while the red and purple flower solutions don't really seem to have done much of anything. Over in the split stem cups, the red-blue flower is showing only blue and exclusively on the side of the flower connected to the blue solution. The yellow-red flower is showing almost exclusively yellow, with the red only just barely showing through. The yellow-blue flower looks a little more evenly absorbed, though strangely not as well as the blue and the red-blue flower. Looking at all 3 split-stem flowers in the first hour, it is clear that the flowers seem to absorb water and nutrients exclusively from the solution below it. So, 4 hours after the start of our experiment, the whole stem green and blue flowers continue to bring more and more color to their petals through the transpiration process. Red is just starting to mark the tips of the petals, and the purple has a center that is strangely tinged green, while the petal tips are getting small stripes of blue. Now, I'm no master of color theory, but none of that makes a whole lot of sense. Checking out our red-blue split stem, the blue dye continues to color its half of the petals, while the red dye is just starting to show up on its half of the flower. The yellow-red split stem is looking more equal in terms of how much dye has been transpired, that has gone through the transpiration process. It also does a really good job showing just how absolute the separation is between colors. And finally, we have our blue-yellow split stem, in which we see the blue continue to creep through the petals while the yellow continues at a much, much faster pace. 8 hours in, and our blue and green whole-stemmed flowers continue to deepen in color. While red does as well, it just does so at a drastically slower pace. Meanwhile, purple doesn't seem to have changed in the last 4 hours at all. It's just some blue along the petal edges and a weak green tint in the center for some reason. Meanwhile, the red-blue split stem seems to mirror the whole stem, with blue being vibrant and red slowly but surely moving through the petals. What is interesting is that the single petal at the top of the red-blue seems to have turned a type of purple, suggesting that maybe there is in fact a tiny bit that absorbs from both halves of the stem. Strangely, neither the yellow-red or the yellow-blue multi-stem flowers seem to have changed much in the last 4 hours. Over the next 16 hours, not a whole lot has changed. The green and blue whole stems look to have about the same amount of color as before, and purple seems, if anything, to have turned a little bit more blue. The only exception seems to be the whole stem red, which, as we observed previously, is slowly but surely using the transpiration process to get the red food dye onto the leaves. After 24 hours, it's finally looking about as vibrant as the blue. Now, before we get into the multi-stem flowers, I wanted to explore a little bit more about what is going on with the purple flower. So, naturally, I cut it in half. So inside the stem, you can clearly see how much red seems to have gotten stuck in the xylem. This suggests that a lot of the molecules that made up our purple food coloring, which again also includes the color green for some reason, were just too large to make it up and through to the petals. In order to get a decent comparison, I decided to cut open one of the flowers that had no problem coloring. In this case, I chose our blue flower. Note that comparatively there is very little blue in the stem. This seems to confirm that even if the molecules in the purple food coloring aren't too big, something is keeping it from getting through the xylem. Now, moving on to our split stems. First, our red-blue, which honestly hasn't changed much. The red dye doesn't seem to have interest in getting into the flower, but we do have that fun dark purple petal, which is something. The yellow-red has taken after the whole stem red and spent the last 16 hours pushing the red dye through the xylem and creating what is probably the best looking of our split stem flowers. Unfortunately, even with the extra time, there doesn't seem to be any obvious blending between our color solutions. Finally, looking at our yellow-blue split stem, we can see that the blue part of the flower has gotten much brighter. What's more, we can actively see some blending on one of the petals. While not quite as obvious as purple, it shows that somewhere in the xylem, our colors are meeting and mixing together. Throughout our experiment, we could clearly see how transpiration took our color solutions and brought them up the xylem and onto the flower petals. However, the sheer number of colors we used, combined with a lack of repetition, leaves us with a lot of unknowable variables. But what worked with the whole stem blue worked with one of the split stems, but also didn't work with the other. Come to think of it, that is exactly how it worked out with our reds. Still, watching a flower, especially one that stem has been cut in half, continue to metabolize nutrients and water shows just how awesome the transpiration process is. Should I have put a whole stem flower in a yellow solution in order to compare it to our split stem? Yes. Should I have limited myself to 2 or 3 colors between the whole and split stem flowers in order to gather better data? There is no doubt in my mind. Unfortunately, I'm out of flowers, so until next time, I'll have to leave the better science up to you.